DNA computing is very important, but if you watch my playlist about theory of computation, you can appreciate the beauty of DNA computing much better. So I'm explaining these two important books about DNA computing, and there are very few books about DNA computing because it's a very young research area, and still it is at its infancy. So we have some molecules, A, T, C, and G, and so think about these as some molecules, because we are just looking at it from the perspective of from the computational perspective, from a computer science perspective. So these A, T, and G and C, as you see, A and T match together and G and C always match together. So it's like a code. It's like a matching. And this DNA strands allows a natural parallelism. And you can do massive parallel computing because you can do many things. Because if we have a copy of DNA and we can create lots of copy of DNA, I will show you how. And so you can do, it's like having multiple computers or multiple processors working in parallel. And this important theory of Watson-Crick complementarity refers to the fact that, for example, A and T match together, C and G. So you can, because there is always a match, for example, consider just a byte in computer systems, 0, 1, 0, 1. If there is an opposite thing, 1, 0, 1, 0, 0, 1, so knowing one of them is enough to understand the other one. So, and these topics are really related to information theory as well. And nature wants to reduce redundancy. And all of these codes are very important. You know, DNA, from DNA, your body creates RNA, and from this, it creates protein. And so all of the things that is happening inside your body is computation. And so A binds with T, G binds with C. This is the thing that is very important in all of the algorithm that will be developed. And you know, DNA computing has many applications. For example, future storage systems are not hard disks, they are DNAs. And you can compress lots of information inside DNA. With some algorithm, you will see how. So classical theoretical computer science, if you watch my playlist about theory of computation, is all about regular languages, and then you go to context-free languages, you implement it with push-down automaton, and sometimes languages are not enough, you go to context-sensitive, and sometimes, and the outer layer is Turing, you implement it with Turing machine. Regular languages can be implemented with finite state machine. Context-free grammar can be implemented with push-down automaton. So you see all of those things, especially Turing, which is very, very general thing, and it's very hard. Turing is all about rewriting operations. So the only thing that you are doing, the only action that you are doing is rewriting operation. But in nature, we have very different actions such as cut, paste, adjoining, insertion, deletion, and I will explain all of these actions. And all of these actions are happening inside your body at each instant of time and in all of your cells. So it's all of human body is just computation. So what is ligation? I will explain this ligation, but DNA ligation is joining two DNA molecules. For example, you see A is matched with T, and so it becomes this. A is matched with T, and so it becomes this. So it's, but I will explain it in future. And or we can represent it like this one. Your DNA could be like this. And so these are complementary sticky ends. And the reason that CG pairing is stronger than AT pairing is just because 3 hydrogen bonds is stronger than 2 hydrogen bonds. And this is a double helix. You see, it's a very stable chemical thing that everything is created from this. All of your protein, everything is. So if this code is not correct inside your body, because of you have eaten lots of chemical things or some other things that is not good for your health, those chemical things edit your DNA, and so you suffer from lots of diseases. So there are many ways for measuring the length of DNA molecules. For example, in a given time span, the small molecules will travel long distance along the ones. So we understand if it takes time, if it takes too much time, it's shorter. The length of your DNA of that particular part of DNA is small. And or you can even increase, you can lengthen your DNA with some operation. You can see all of these are chemical operations that are happening. And you're lengthening your DNA. Or sometimes you're doing the opposite, you're shortening it. You can make it smaller and smaller. So these codes, using some chemical operations, you will see that they will shorten and shorten. And the other action that we do the body, the nature does, is cutting. For example, the restriction enzyme will bind to DNA. So a particular restriction enzyme will bind to DNA, binding here, and it cuts that at a specific recognition site, and sometimes out of this recognition site. So it will cut this bond between adjacent nucleotides in such a way that it generates the OH group and here at end of the one nucleotide. So this is the cutting DNA. As you see, cutting from here, you will see you can have 2 different chemical structures. Consider it in very complex structures. Or you can have multiple cutting. Instead of just cutting from one part, you can cut it from multiple areas. So you have more complex chemical structures. Or you can do linking. This is another important action that you can do, is linking. You have 2 complex structures. You link it using some, for example, hydrogen bonding. You have this complementary base pairing.